Hello, everyone. Welcome to the Monash Mad Science Talk Show. Da-da-da-da-da-da. Today, we are joined by mad scientists Fadden, Huishan, Samantha, Chunkit, and Olivia on their proposal to investigate the effects of temperature increase on a species of coral reef, the Acropora grandis, in the Australian Great Barrier Reef. Fadden, you now have the floor.
Thank you. I will start by introducing to you the significance of the Great Barrier Reef and why it is so important to study the effects on coral reefs. Firstly, the Great Barrier Reef stretches about 2,000 kilometers off the coast of northeast Australia and covers an area of about 400,000 square kilometers. Two things make the Great Barrier Reef so unique for the study. Firstly, it has one of the largest coral reef system with about 4,000 square kilometers of coral cover and almost 3,000 known coral species to this date. Secondly, the Great Barrier Reef is one of the least threatened coral reef as it is far from major cities and is majorly protected by Australian law. However, in spite of this, coral cover has reduced from 28% to 13.8% in 2012 for the past 27 years. This was mostly due to increased water temperatures, coral disease, tropical cyclones, and terrestrial runoffs. The corals are also experiencing thermal stress, increasing calcification rate by 15 to 20%, decreasing coral recovery, growth, and reduce its tolerance to bleaching. Although bleaching mortality is the lowest compared to other regions, this is certain to increase as temperatures increase in the Great Barrier Reef. Coral reefs are such an important model to use in the study as it provides so many services to the local marine ecosystem, such as shelter, habitat, and breeding ground. The coral reefs can also act as a natural barrier from tsunamis and prevent coastline erosions. We can't also dismiss how beautiful a coral reef landscape can be. Next, we will dive a bit deeper into the importance of this study and why it's worth exploring.
In the context of Acropora grandis related studies, various studies such as the effect of high temperature on heat shock protein, relationship between bleaching and hydrological factors, which found that severe bleaching of Acropora grandis occurred when ambient temperature is high, and the survival and growth after transplantation in the Andaman Sea have been studied, which showed that the survivorship of Acropora grandis after transplantation was 87%. However, no direct study of the effect of temperature increase on the population ecology of Acropora grandis has been conducted. By considering the fact that no study has been conducted regarding these variables, the research question in this study is as follows. What effect does temperature increase have on the population ecology of Acropora grandis in the Great Barrier Reef? By answering the question, the research gap in the literature can be filled. This study also aimed to extend the existing knowledge by empirically evaluating the effect of temperature increase on the population of Acropora grandis. Now, we will be talking about the taxonomy of Acropora grandis.
Acropora grandis is a species of colonial staghorn coral. It is a species with multiple branches which inhabit subtidal zone from 5 to 25 meters deep. Acropora grandis are known to tolerate wide variety of environments, but are mostly found in shallow reefs and lagoons. They are normally reddish-brown, blue, or green in color and have a paler color at the tips of the branch. This picture shows the life cycle of a coral. The life cycle of Acropora grandis has an average generation length of 10 years with average maturity age of around 8 years. Acropora grandis practices synchronous spawning and the gametes are carried and dispersed with the help of sea currents. With the influence of sea currents, Acropora grandis population are distributed in areas such as the Red Sea, Gulf of Aden, Sea of Japan, Sea of China, and basically areas in the central of Indo-Pacific and of course Australia, which is the Great Barrier Reef as shown in the picture. Next, let us know more about the current population trend of Acropora grandis.
The population trend for Acropora grandis is currently decreasing, and this is due to various factors such as climate change, rising sea temperature, and ocean acidification, which are all the result of disturbances from human activities. Acropora grandis has low resistance and tolerance towards bleaching and diseases, which could cause negative effects for the coral. As for the disease outbreak, It is due to rising sea temperatures, which will affect physiological and biological mechanisms of the coral, which will further impair the defense mechanisms of the coral. As for coral bleaching, Acropora grandis is one of the most affected species in these bleaching events, and a type of symbiotic algae called zooxanthellae, which is responsible for both providing energy and color to the coral, will be expelled from the coral under increasing temperatures, as zooxanthellae will produce chemicals which are toxic to the coral under increasing temperatures. This will cause coral bleaching and the death of the coral, which will cause a change in coral populations, such as reduced growth rates, decreased reproduction rates, increased susceptibility to diseases, and raised mortality rates for the coral, which will then further affect other living organisms which are dependent on this living coral, such as fishes and invertebrates. The reason why we chose Acropora grandis for our study is because it is one of the most affected species in these bleaching events, and it is also one of the most common coral species due to its tolerance to a wide variety of environment. And by understanding how temperature increase negatively affects this tolerant coral species, we will also be able to have an indication of how serious and dangerous temperature increase is to all living organisms in general. Acropora grandis is also easily identifiable due to its unique branching pattern, as shown on the figure on the right. It is also found in shallow water regions, which makes it easier to collect samples for our study. Next, we will be moving on to the methods and materials used for investigation of our proposal question.
Now I would like to talk about the materials and method to test the effect of temperature increase on the Acropora grandis in the Great Barrier Reef. According to journal article and literature, we know that Acropora grandis grows in a clump distribution. Therefore, we will do an in situ experiment to measure the bleaching matrix by categorized bleaching intensity. You can refer to the figure on the right, so we assume the Acropora grandis grows in a clump distribution around the corner in our defined quadrant. They will be divided into 8 populations, and we will collect our data every month for a year span of 12 months. Every month, we will go to the site to calculate the bleaching area coverage in meters squared. Then, we will also set up a thermometer near to each population to detect the temperature changes every day. So for example, in January, we go down the Great Barrier Reef to obtain the bleaching area coverage and constantly record the temperature every day to get an average temperature for the whole month. This same action will repeat for 12 months. After 12 months, when we get all of our data, we will plot a bar chart for changes in bleaching area coverage each month against temperature, so that we know how does the temperature affect bleaching area coverage in Acropora grandis in the Great Barrier Reef.
If any of you would want to know more about this research topic, here are the references you can refer to. And that concludes the end of the Monash Mad Scientist Talk Show. Thank you, Mad Scientists, for joining us, and thank you for listening. Da-da-da-da-da-da.